We've now going to move to a very quick Q&A session. So I'm just going to invite all of the session speakers back up on stage, and we will go straight to any questions that we have from the audience. Harriet.
Thank you. I'm Harriet Bailey, International Manager at the Australian Antarctic Division. There's been a lot of talk about science today, obviously, but there's also been a lot of talk about technology, and that's a really different aspect. And this also links a bit, I think, also to the panel we had before. What are we doing to make sure we've got the right technologists who are working with us, and how we train them up?
You're a little bit premature, Harriet, in that our very next speaker is going to introduce our technology program that could probably answer a lot of that. But it's a great question, and the two things go hand in glove. Really, our technology and our science branches are intimately linked and really informing each other frequently. And so I'll let Phil talk a little bit more about the specifics. Dave.
I have a question. That's okay. Question for Leonie. So you were talking about how rapidly krill DNA degrades. Is that the same for all species? Do they all degrade at the same rate, or?
Yeah, well, it depends on a lot of things. It depends on what environment you're in. For example, warmer temperatures usually lead to faster degradation. There were experiments with different fish species in the exact same environment, and their eDNA degraded at different rates. So it depends on a lot of things, and just from what we did with krill here, I wouldn't want to make any predictions of, you know, any other species in a different environment or even the same environment. It pretty much needs to be tested for each species separately.
Thanks, Leonie. Nathan.
Yes, thanks for those. I think this question's to either Aleks or Jonny, and it's to do with the fact that no one lives in Antarctica. So the question is, how do we adapt, and maybe Barbara as well, how do we adapt and support those species in a ongoing changing climate?
Thanks, Nathan. I think we've got some great... No, seriously, thank you. It's a good question, and I think what it does is it allows me to remind you and others that we actually have some really clear instruments in place that allow us to put in place mechanisms for protection and conservation, I think. You know, the Antarctic Treaty, the Protocol on Environmental Protection of the Antarctic Treaty has got mechanisms in place. The Committee for Environmental Protection and CAMLR have got the mandate and the power, if you like, to make decisions that can protect these species. And I think, from my point of view, where I see the most value in my science, and we'll talk about this in Guy's-led symposium or discussion later this afternoon, is when we work together with policymakers and then our national delegations, and I say our because we're working cross-country here, to actually get those right decisions made. And I think decisions in those forums, difficult as they might be sometimes, are actually the key to the protection and the changes and managing the changes that we're seeing. That's a short answer. I could talk to you about it for longer, but for now, that's my first cut at it.
Thanks, Aleks. I know Dave's trying to wrap us up, but I have a quick question for... Oh, you have another question online? Go for it.
So we have a question online from Gwill Price. Question for Leonie. Email. Is there research at the AED going on into using eDNA for quantification of species in the Southern Ocean?
So that method I've been talking about, that targets krill, that is quantifying the krill eDNA in a sample. Now, whether the amount of krill eDNA actually relates to how much krill there is an additional question. So krill eDNA abundance that we can detect, whether that actually relates to abundance of krill detected with other methods. That's the next step that we're working on. But yes, there's different genetic methods. Some of them will allow you to quantify the eDNA of a particular species. Others are more present absence. But yeah, the krill one is the only one at the moment that we're doing to really quantify. But then there's a lot of other work that is detecting a whole range of other species, including penguin or other predator diet, as we can tell from this, what's left in the scats, what DNA we can detect in the scats. So there's a lot of different work happening.
Thanks, Leonie.
Nicole, do you mind if I just add something to Alex's answer in response to Nathan?
Go for it.
Just an opportunity. So Nathan, I noticed part of your question specifically related to people in Antarctica, and I think that's a really good question because as you could see from my talk, people are concentrated into particular types of areas in Antarctica. And when we talk about marine protection in Antarctica and the proposals that go through the CEP and CAMLR, they tend to focus more on the Southern Ocean and offshore regions. And we already have a system of protection for terrestrial areas. So I think coasts and those coastal environments are actually an overlooked area in terms of conservation and protection, and something that is worthy of consideration, particularly around the fact that people are concentrated in these areas and there might be some very sensitive habitats in these coastal areas. And so just being aware of those and being able to manage our presence there in relation to that will go a long way, as well as maybe then also thinking about marine protected areas in some of these coastal ecosystems. So not just the Southern Ocean, but incorporating that whole transition right up into the terrestrial.
Thanks, Jonny. You actually segued into the question that I had for both you and Aleks, which is, we heard from the Minister's words this morning, her focus on the East Antarctic MPA, and I'm just wondering if you could make a comment on how fit for purpose our marine science program is in terms of delivering the science in to be able to inform the design of an East Antarctic MPA.
Thanks, Nicole. I think the short answer is that it's very fit for purpose. It's understanding what we have in those ecosystems is absolutely fundamental to making a strong case to protect them. Now, we don't need to know about everything that's there, but what we do need to know is have some broad indications of where we have vulnerable species or where we have important species, areas of biodiversity. And I think the work that we're doing gives us that information and actually provides the strongest possible justification for getting MPAs, like the East Antarctic MPA, up.
Thanks, Aleks. It's exactly what I wanted to hear. Jonny, do you want to add to that?
Oh, just very briefly that, with the knowledge of the partnerships that we have across the SRIs and the AAPP, I think we've really got it nicely covered, all those different habitats and environments to be able to inform that decision making around area protection.
Great. David. And David might be our last question for the session so that we can try and regain some time.
David Suter, Chief Research Officer at the Australian Institute of Marine Science and soon to be AAD. For Aleks, you made the point in your talk that you had heaps of data, and you just also made the point that you don't need to know about absolutely everything. With the sort of soon-to-be established East Antarctic monitoring program, I'm just wondering if you've got a sense of the process or a framework to think about what are the actual critical data, the critical indicators that you need to measure going forward.
Thanks, David. I think what we need to do first is to understand what we're actually doing. So at the moment, for example, we are, the collective we, I'm not exactly involved, but there are people in the Antarctic Division and elsewhere that are actually trying to understand what we're doing both at the AAD and more broadly across the Australian Antarctic Program and more broadly across Antarctica. I think that's a really good starting point. I think then we look at our levers and our drivers, if you like, our triggers for monitoring. We have some legislative and statutory obligations. They're obvious ones. But we also have things like threatening processes that are going to, if you like, elevate the priority, or threatened environments, threatened species that may well elevate the priority. And so I think starting with an audit, going through a process of understanding what our key drivers are for monitoring, then looking at the if you like, the processes that might be affecting species. I think through a process like that, we'll actually start to get pretty close to understanding what's essential, what's important, and what can maybe be sort of left on the back burner for the time being. So that's a very short snapshot of a probably fairly long and involved process. Thanks.
Thanks, Alex.